All right. Any questions before we start where we left off last time? All right. Let's see. Okay, Robin, you said you couldn't see any comments on the paper. Did you open it in Microsoft Word? Because you often can't see, I did it in track changes and using the comment function in Word. But if you want to stay, if you can stay a couple of minutes after class, we can try to get that figured out. All right, thanks. And if anybody else is having those issues, please just let me know. All right, so I will share screen. So we left off, as you may remember, talking about counting things. And what we had talked about were culture-based, live, viable based counting approaches. Okay. So that's really one method, right? And as we read about before, there's limitations. As we've talked about before, there's limitations, right? And so what we're going to finish up with today is talking about direct counts, right? And this is really the other side of the coin in terms of the enumeration process. And this is how we discover the Great Plate count anomaly. When we count using direct counts, it's usually much larger numbers, often at least one order of magnitude higher, if not more, right? So if you use that ballpark number, which is just a ballpark, remember, of like less than 1% are culturable in most environments, that means it's 2 orders of magnitude higher, 100 times the numbers at minimum. So the way these work is what many of you have already done before, right? You've already counted something under a microscope at some point, right? Maybe in a hemocytometer, like a counting chamber. Or what we typically do with bacteria and archaea is we will filter samples down, and often the filter will have a grid. And so you'll count squares, you'll count boxes. What you can see in the background here is fluorescence microscopy. And so you can see these individual cells, right, that are lighting up through the use of a stain. And so epifluorescence microscopy really makes it much easier to see the individual cells. Think about light microscopy. You usually need to use a stain to get the cells to light up. But even then, it can be hard to really count. And so it turns out that fluorescence makes it easier. Now, some cells automatically fluoresce or autofluoresce when they're exposed to light of a certain wavelength. So for example, if you shine light on a phototroph like a cyanobacterium, the chlorophyll molecules inside the cells will fluoresce back at you. So if you shine green light, they will fluoresce in kind of a reddish color back at you. Okay. Methanogens within the archaea are similar. A different set of wavelengths will excite different pigments. It's not a photopigment in the case of methanogens, but it's something that will light up. You can also artificially light up cells, and that's what's shown here. This is acridine orange, which is a very traditionally commonly used stain, which turns cells this kind of yellowish greenish color, or orangish and greenish color. Today, however, we usually use DAPI. And if you remember the Oxenwright paper, that's what they used to count their cells. They stained with DAPI. Both of these are nucleic acid stains. Acridine orange will stain both RNA and DNA. DAPI is primarily for DNA. So in a bacterial cell, it's going to stain the nucleoid, right? And it'll stain our cells too. So in a eukaryote, it would stain the nucleus and the chromosomes. Now we're going to learn more about another approach next time in terms of counting quantitative measures of fish. It's probably not going to get there today, but we'll see. So epifluorescence microscopy relies upon these fluorescing molecules, which we call fluorochromes. And all that is a $10 word for something that glows, right? And so again, it can be autofluorescence. And which groups did we say do that?
Phototropes and methanogens.
Exactly, thank you. And then using those dyes, these are a couple of additional dyes in addition to the acridine orange that I mentioned and the DAPI. There's actually many different kinds of dyes and they fluoresce at different wavelengths. And so you can get creative in how many things you can stain at the same time using that. The one thing that all of them have in common, though, is something called Stokes shift. So or I guess Stokes was the researcher's name, one of the early microscopists who looked at fluorescence. And what they found was that the emission wavelength is always longer. So that's shown here. This is the acridine orange fluorescence spectrum. So what you can see is if you shine a light of about 470, 480 nanometers on an acridine orange cell, then you can see that it fluoresces. But what wavelength is it fluorescing in? What's the peak fluorescence there? Who can interpret the graph? So there's a shift from about 480 to what? Can you guys see the diagram?
520? Like 520?
Yeah, like 520, 530, something like that. Okay. So that's red shifted. This is a term that you'll hear used in the fluorescence community. This is the blue and this is the red, right? Roy G. Biv backwards, right? So Roy G. Biv goes this way. So you're right shifted, you're red shifted. Okay. Another thing we need to worry about when we look at photochromes or fluorochromes is fading. If you have an autofluorescent cell or a dyed cell and you start shining photons of light on that sample under the microscope, over time they bleach or fade. Okay. And so you just have to be wary of that. There are some chemicals that you can use to try to minimize that, but you also need to be quick when you count things. Now, the advantages of direct counts, as we've already mentioned, is that you get much higher numbers typically. They're also not environment specific. Okay, you can do a direct count from anywhere, although doing it from an aquatic sample is usually easier than a soil sample. Why do you think that would be? Why is it harder to do a direct count with a soil sample? What do you think?
Because it's solid media.
It's solid, right? And what did we say is often the first step we're going to do before we count things?
Filtration.
Exactly. So if you're going to filter it, then what you need to do is take your soil, add water or some other medium, and resuspend the cells off of those soil particles, and then probably let the soil sediment out, and then you could, sort of pipette or transfer those cells over onto a filter. A little bit challenging, but it's definitely doable with some protocols. There's also a correlation with biomass in the sense of the more cells you should have, the greater the biomass, right? So it should be a positive correlation there. Now, drawbacks are that if you stain something, typically, what do you do before you stain that sample? We mentioned it last time. When we were talking about our lake sample, I think one of the groups said they would do this too.
Is it like fixing the microbes?
Right. So if you fix them with formaldehyde or formalin, right, they're dead, right? But they will still light up. They still have DNA, they're preserved, And so what happens then is you're counting them, but you don't know if they were alive or not. So one of the disadvantages compared to the viability, with a viability count, you know they're alive, right? They've got to be to grow. But there are some stains. So there's this live dead backlight system that was developed by a company called Molecular Probes that does quite well. So it will light up a different color if it's a dead cell versus a live cell. And then debris interference, which particularly would be an issue with the sediments or the soils, can get in the way. If you have like gunk, whatever you want to call it, debris, schmutz, whatever you want to call it, it's things that would make it hard to see the cells potentially. So you need a clean sample. And if we killed them, we can't go any further in terms of like culturing those organisms, right? We can just count them and then we would dispose of them. All right. It's also laborious. At a school like Cal State Long Beach, if you work in a micro lab, they might have you count cells under a microscope for hours and hours and hours. And, you know, that's sort of your research project. But it's laborious. It takes hours and hours and hours, and you have to be trained on how to count properly and consistently, right? That's the key. You need to do a lot of counting so that you know that you're reliable. All right, so another way to get around this is to automate the counting. So there are digital counting programs, software, so you can purchase image analysis software, which will count all those lit cells, particularly if you use fluorescence microscopy like we talked about, but you need a clean sample. Otherwise it won't count it properly. And it can be, these software programs can be expensive, although I think they're cheaper now. There's a free one called ImageJ from NIH. Some of you may have used it before, but it's not automated. So flow cytometry is now the more commonly used, and we actually have some flow cytometers in the department. And this is where you take cells, you resuspend them in a solution, and you push those solutions down a tube, like you can see here, where the sample goes through this, and you either have autofluorescence or you stain them. And as they pass through this tube, they'll go past a laser detector and it will fluoresce, or it'll flash every time a cell goes by. And so this can be a really powerful way to very rapidly count cells. It's quite expensive to get that, but if you have the machine, then it can really save you a lot of time. And so, has anybody used a flow cytometer if you work in maybe a research lab? They're pretty useful. All right, so that is the enumeration methodology. Any questions about that? All right, so we will move over and start talking about molecular sequencing. So this lecture and the first part of the next lecture will be working on talking about molecular methodologies. Today will be a lot of sort of PCR-based approach, and then some of the quantitative methods will be next time. We might get to some of it this time. But basically, what we're going to do is what we already read about in the Oxenwright paper, right? Instead of culturing organisms, which they also did, you can go directly to the environment and you can take that.
Dr. Dillon?
Yes.
Sorry to interrupt. Did you upload this for us?
I thought I did. Hang on. Maybe I didn't. Let me see. Maybe it's not visible. Let me run and look real fast. Oh, apologies. It's not visible. I hate that it defaults to being invisible. Sorry about that. All right, you should be able to see it now if you can log in to Beachboard. All right, let's try again. Thank you for letting me know. Oops. Okay. All right. So can everybody see that? All right. So the first step, as we talked about, as we read about in the Oxenride paper, is the use of DNA extraction. And so I wanted to just give you a little bit of insights. They kind of glossed over it, or you may not really have understood when you read that. So what you need to do is burst open the cells. So you can either use enzymes like lysozyme, okay, which works particularly well with gram-positive cells, or you can use detergents, right? So SDS is a very commonly used detergent. But just like the detergent you use on your dishes, right? It's going to break down lipids, right? Like the fats in your food. Here, we're breaking down the lipid bilayer and bursting the cells that way. You can also do it mechanically, though. So you can freeze thaw, and that's what they did. Anybody remember how they froze their cells in the Oxenride paper?
I believe it was liquid nitrogen.
Exactly. So you can take a sample, drop it in liquid nitrogen, it'll freeze really quickly, pull it out, drop it in a hot water bath, and boom, those cells will just burst right open. Or you can mechanically do it using a bead beater, which is a commonly used. And basically, these are little tiny zircon beads that you just shake the heck out of it and disrupt the cells that way. After you lyse the cells, you need to clean them up. And this was also described in the Oxenride paper. Right, so you can use something like proteinase K to get rid of those proteins again. And then you can do an organic phase separation. Now, in the old days, you used phenol and chloroform, but those, as if you've ever experienced either of them, are really nasty chemicals, right? And so these days, we typically don't even do this. We just use a kit. So instead of having the phase separation be in two different organic solvents, what you do is you just stick your sample, your DNA sample, onto like a membrane, and then you can do washes and lysis steps and all these things with it, and then you just elute it back off. Okay. All right. So Robin, you asked the question in the chat, why were the Neutronococcus difficult to break open with the freeze thaw method? And that raises an important point. Not all cells are created equal. And we don't always know why one lyses more easily than another, right? You can use the same method. You can even use the same method that has worked for a species of Neutronococcus in the past. Should work again, but sometimes it doesn't, right? So these methods generally work. And so, you might want to use the bead beating method in that case instead of using, now typically with a pure culture, it's easier to lyse things. With an environmental sample, you may have like particles, debris, other things in there aside from the cells, right? And so they can make it a little bit harder. So bead beating, I would say, is more commonly used with that. Freeze thaw is used for both, but we don't always know the answer to your question, Raman, I guess, but it's something that you've got to figure out. And if you try multiple methods, this can be a way for you to figure it out. All right, the other thing you want to do is clean up your sample. And with those kits, you don't typically need to do this, but if you need really pure DNA or RNA for that matter, a cesium chloride gradient is the best way to purify it. So if you're going to do like a genome sequence of a bacterium, then running it through a cesium chloride gradient would allow you to get rid of everything else except the DNA genome for that. When we're going to use our sample for PCR, which is very common in the methods we'll be talking about today, one of the things we're worried about are contaminants or inhibitory PCR inhibitors. And you remember this term humics. Where did we encounter that in our habitats? Do you guys remember? So we talked about humic acids as being particularly prevalent in soils. So remember, those are those high molecular weight organic molecules that polymerize from decaying vegetation, plant matter in the soil, which is what makes those soils really rich, mineral rich and carbon rich for plants to grow in, but they also can inhibit PCR. And so what we often do is add chemicals. So bovine serum albumin, which is just protein, right? This can be added to a PCR tube, or you can add this polyvinyl prolidone or there's some other chemicals that are sometimes added, and that can help remove or sequester the inhibitors as you try to run the PCR with it. And then another thing that can be done to purify just the nucleic acids is to precipitate them. Now, if you're using a kit, you don't have to do the precipitation step in a tube. You just add alcohol while the sample is on. These are little like spin filters that you use in these kits. But if you do it in a tube, then you would be pelleting or precipitating out your DNA in the bottom of a tube. And then you get rid of everything else and you can resuspend it in a small amount of sterile water. Okay. So this gives you pure nucleic acids. Primarily what I'm talking about here is for DNA, but you can actually get RNA if you change the pH of your phenol. Okay. All right, I'm not going to go over PCR in this class. You guys have all done PCR in some sort of intro bio lab, I hope. Or if not, go on this YouTube site. I think it's still, I didn't check it this morning, but just Google YouTube PCR and you can see how it works, right? But basically, we're making lots of copies of our DNA using Taq polymerase or some other similar polymerase. And this is just what we read about in the Oxenwright paper. We do have some caveats to think about here, though. And this pertains to the entire process I just described, extraction biases. So, and this kind of gets to your question, Robin, is that why are some things more difficult to break? Some cells are more difficult to break than others. Well, if you have an environmental sample that you don't know what organisms are in it to begin with, guess what? You have no way of knowing if there were Neutronococcus in there that were hard to break open. And so we just need to understand that we need to try to use as good an extraction procedure as possible, but we have to have the caveat that we could have had incomplete lysis. Or on the other hand, we may have extracted DNA and it may have degraded. So you've always got to wear gloves and work in sterile tubes and benches and everything when you're doing DNA or especially RNA work, which is very delicate and falls apart really rapidly. So degradation by DNases and RNases are really an important consideration to take into account. I think more seriously is PCR bias. So we have these primers. And remember the Oxenwright paper, what kinds of primers did they use for that study? What were they
16S
targeting? 16S ribosomal RNA, and what group were they targeting?
Archaea.
Exactly. So they knew in advance they wanted to target Archaea, which by the way, was one of their assumptions, right, that they made, right? They assumed it was all going to be Archaea, and so they used those primers, right? And that's all you should get out of it if you use those primers. There are so-called universal primers that should amplify both bacteria and archaea. There's even some primers that may amplify both 16S and 18S, although that's sort of tricky to do. But let's say we have a set of samples and we want to amplify both the bacteria and the archaea from it. We can do that. However, we know, and there are some very sort of like interesting type cases that have shown that the primers are not perfect. The way you design primers is you look at gene sequences in databases and you try to find places on the gene that are conserved and the same across all of the species you want to amplify, but nothing else. So for example, the archaeal primers that they used, you want them to amplify every archaea, but no bacteria, right? So you have to design them to target a specific space that doesn't vary with archaea, but is different than what bacteria would have in that part of the gene. Does that make sense? Well, so even with these universal genes where you're trying to get everything, right? It turns out there are some fairly high profile cases. So there was this really interesting archaea called nanoarchaean, which was discovered now a long time ago. This was back before I even had this position at Cal State Long Beach. But it was an interesting example because this was a symbiont. They discovered this nanoarchaean, which had another archaea that lived in co-culture with it. And both of them needed each other to survive. So this is an example of those culture issues and how symbioses can be important. So they had this two-part symbiosis in the tube, but when they used the standard primers to amplify them, they kept getting one thing out of it. And so when they were eventually able to sequence the 16S gene using different primers, they discovered that the so-called universal primers didn't target one of the two members in that tube. So we can do our best to try and, do as well as we can, but primer bias does exist. And the problem here, like with the extraction bias, is you will never know what you're missing because you've never seen it before if you're using these primers, right? Now, the good news is now we can do whole metagenome sequencing, which doesn't rely on primers. And so we can now kind of get around this issue in some ways. And I'll talk about those approaches later today or early next time. Another caveat of PCR is that it's not truly quantitative. So, when they were counting clones of like, oh, we had 12 halorubrum clones, et cetera, they couldn't then say, oh, that meant that there were 12 halorubrum cells in the environment, right? Because PCR is it's an amplification that occurs, but you don't know how many cells you start with. You don't know how many 16S gene copies they have, and that can vary from one organism to another, by the way. And the number of cycles that you use actually matters in terms of getting the cells to amplify, the genes to amplify, I should say. And the longer the number of cycles you use, the more product you'll get, which can seem good. But remember, as with a logarithmic growth curve, a PCR reaction will be very similar. It'll have a lag and a log and then a stationary phase. And if you go too long out into the stationary phase, you can actually, the Taq polymerase can make mistakes. So we need to have a balance here. Incidentally, this is of interest for us with COVID because when you go to have a COVID test, many of you, like myself, have probably had to go have your nose swabbed for a COVID test over the last 9 months. And when you do that, they take that swab and the way they do your test is via PCR. So they use CoV-2 specific primers to amplify and determine if there's RNA, it's an RNA virus, remember, So they use target that gene to get it to amplify in a PCR. And what they have found actually is that you can end up with false positives if you run the cycles too long, and false negatives if you run it too few cycles, because you have to get enough of the PCR product as it goes up, up, up to be detected. So this is relevant for human health as well as our environmental samples as well. Any questions about those caveats? Does that make sense? Am I going too fast with that or? Okay. If not, you can always watch the video. So and really what I will say is if you want to count things to be quantitative, you need to go back to the environment. And so we're going to talk about some methods, particularly FISH, which can be used to count cells in the environment. Okay. And you can get PCR results, 16S genes from a sample, design specific probes for that, and then go back and check. And we'll talk about that in a few minutes. All right, so let me see if I can get this to go up. Come on. Okay, let's have our first poll of the day. All right, so which of the following would be caveats or limitations of PCR-based molecular diversity approaches? A, PCR is quantitative. B, not all cells are equally extractable. C, even universal PCR primers don't amplify all microorganisms' 16S genes. D, both B and C, or E, all of the above. So I will launch the poll. Go ahead and choose your answer. All right, log your answer if you haven't already, please. Okay. All right. So about 2/3 of you put D and the rest put E except for one person. Okay, so let's take a look at that. All right. So the question is PCR quantitative? That's the first one. What did we just say about PCR? Is it quantitative?
It isn't.
So the caveat is that it is not quantitative. So that was a little bit tricky. So I think I got those of you that put E, I think I tripped you up that way. I will say that these are, I'm not going to have multiple choice questions on the midterm, but these are the kinds of questions that you will expect to see on the midterm in terms of, I will ask you like, what method would you use? And then I will ask you to identify potential caveats or pitfalls associated with those methods. Okay. So it's important to read the questions closely in order to do that. So I think most of you got that B and C were both correct. Not all cells are equally extractable, as we said, and then even so-called universal PCR primers don't amplify them. All right, any questions about that? All right. Now PCR is, this wonderful tool that we have, and it has been adapted, and I'm not even going to go over about half of the methodologies used with PCR, but there are some that are very commonly used in microbial ecology studies. And one of the major ones that you will encounter in this field is asking the question, what is the activity or the expression of a certain gene in an environment? And one way that you can address that is you can do reverse transcription. And who knows where we got reverse transcriptase from? Does anybody know?
Actually,
Say again?
Retrovirus like HIV.
Exactly. So RNA viruses, right? Right. If we have RNA viruses, they're RNA. But if you want your cells to copy their genomes, you need to convert your RNA to DNA. And so we can use the reverse transcriptase enzyme that we got from viruses in this regard. So what we do is we convert the mRNA transcripts to cDNA, right? Which is, it's not the entire genome, right? Remember introns and exons and all that good stuff, right? But what you're doing is you're taking the transcribed portion and you're converting it to DNA. And then once it's DNA, you can just run it through a normal PCR reaction and it's just fine. So this is very commonly used when we want to compare functional gene diversity as well as activity. So this, if you, let's say you did the nitrogenase gene, which is for nitrogen fixation, right? If you wanted to know what the diversity of nitrogenase genes that are being actively used in an environmental sample are, you could take a sample, extract the RNA, do a reverse transcriptase step, and then PCR amplify it out and sequence those genes. You'd be sequencing the nitrogenase cDNA, and that would give you the diversity of nitrogenase producing organisms in that environment. Does that make sense? Now you can also do quantitative or so-called real-time PCR, not to be confused with RT-PCR. It's sometimes the same nomenclature, but let's just call this qPCR for our class. And we'll talk in probably next time about qPCR methods, okay? As I said, there's others as well. Now, I'm not going to go into this in too much detail, but I did want to show you this because it was talked about in the Oxenwright paper. So as you remember, that paper was from the early 2000s, and it sort of preceded the next generation sequencing approaches that we use today. And so they did cloning into E. coli. You remember that? So they took their environmental sample and they amplified, they extracted and they PCR amplified it. And then the next step is to clone it, okay? And the real reason for doing this is that we can separate out unique like 16S gene sequence types. That was why they were doing it in that study. So you have a mixed environmental PCR. You know, when they went to that Saltern or that potassium mine, right? They had their sample, they extracted it and PCR'd it out. But as they found, there's more than one kind of halophile living in those environments, right? And so if you just took the DNA and did a PCR on that and tried to sequence that, it would come out as gobbledygook because there's more than one sequence type, more than one read, right? They each have a unique 16S gene. And so different sequences would have different base pairs at the same location. And so you have to separate them somehow. So cloning was really the original way that we did that. Okay. And so basically what you do is you stick your PCR product into a vector, which is usually a plasmid, and that's called ligation. And then you take that ligated plasmid vector with the PCR product in it, and you put it into E. coli. And we call that transformation. We are transforming E. coli. Now, these are not run-of-the-mill E. coli. These are special, very fragile E. coli where their cell walls have been modified so that they will very easily take up these plasmids. And you either accomplish that by heat shock. So you take an E. coli that normally grows at 37 and you put it at 42 degrees for a couple of minutes, and it will actually slurp up the plasmids inside their cells. You can also zap them with electricity called electroporation and accomplish the same thing. And then basically you take those E. coli and you grow them on a plate. And what we can do is we can grow the colonies, and we need to determine twofold screening for this, right? So what we need to know is, does the E. coli have the plasmid? And does the plasmid inside the E. coli have the PCR product in it? And so the first thing is accomplished by antibiotic resistance genes. What you do is on the plate, you put like ampicillin, which is like, a standard antibiotic. E. coli is naturally not resistant to ampicillin. It will die on an amp plate. But if it has the plasmid, you put a gene in the plasmid for ampicillin resistance. Okay. So the manufacturers have done this already. You buy it from them and it's already in there. Okay. So that's the first level of screening. The second level is we need to know, does the plasmid contain our gene of interest? In this case, the 16S gene. And the way you determine if it has that is through a disruption of a gene. So you guys have all learned about the lac operon at some point in the genetics class, right? Yes. If you've taken genetics.
Yeah.
So the lac operon, right, is a complicated set of genes that's going to, change sugars, et cetera, et cetera. We can take advantage of that actually. And so they put lac operon genes in the plasmid, and they put the gene right at the place. So in the plasmid, before you stick the 16S gene in, the gene is intact. And when we do the ligation step, you get this opens up, and in that spot, inside the lac operon gene, you end up sticking your 16S gene. And when the 16S gene is in the middle of the lac operon, it doesn't function anymore. So what you can do is, when you plate your cells, if they grow on ampicillin, they've got the plasmid. And if they produce a color, that means their lac operon is still functional, and we don't want those colonies. What you're looking for are white colonies, which will have we know it has the plasmid and it has the lac operon disrupted. So we know it has our 16S gene in there. Does that make sense? Any questions about that? So that's a fairly rapid explanation, but now you know how cloning works if you've never learned that before. And so that's exactly what the Oxenwright paper was showing. That's the method they used. All right, so let's have another poll question. So what is the main purpose that we just said for using cloning? Is it to aid in identifying E. coli? Is it separating out individual 16S genes? Does it allow us to see a diverse set of sequences in a single clone? Does it allow us to test for antibiotic sensitivity of bacteria, or E, all of the above? Which of these is the best answer? Okay, go ahead and answer if you haven't already. All right. Okay, we seem to be kind of all over the place. So let's look at this. We have quite a few that put E, all of the above, and a lot of people put B. All right, so this aids in identifying E. coli species. So we talked about E. coli, but for example, in the Oxenwright paper, were they identifying E. coli species there?
No.
No, E. coli, you have to remember, is just a tool. It's like the vehicle for allowing us to get lots of copies of that gene that we can then send off for sequencing. Okay, so we're not really usually interested in identifying E. coli species. So that's not the purpose of cloning. Now B, a lot of you put B. This separates out individual 16S genes. That's absolutely correct, right? We said that what we're doing here, and maybe I didn't make this totally clear, was in the plasmid, when you do that ligation step, we said that we were looking for a disruption of the lac operon genes. And what should happen, and what typically happens, is only a single 16S gene copy gets put into each plasmid. And so let's say we have 12 different species of halophiles from that each of those samples. Then one copy went into one clone, and a different species copy went into a different clone, and so on and so forth. So B is a correct answer. Now let's look at C. What's wrong with the statement in C? It's almost right, but it's not quite. What's wrong with that? Who knows?
There would only be one sequence within a clone.
Exactly. So in light of what I just said, we can see a diverse set of sequences in many clones, but in a single clone, there should just be one 16S gene. Okay, so each clone of E. coli, when that colony grows on the plate, and then usually you pick it off the plate and grow it up in liquid broth, everything in that tube should have the one copy of the 16S gene. Then you can do what we call a plasmid prep, or you can PCR it again and send that for the sequencing. And everything should have the same sequence of the 16S gene that was in there. Johan, do you have a question?
So what does individual stand for in this? As in, is it the bacteria? Is it the plasmid or the E. coli?
For B?
Yeah, for B.
So that I meant the genes, right? So each unique 16S gene. So what we're doing is let's say we have 10 species of halophiles in one of those samples from the Oxenwright paper, right? You have a mixture there. And there may be more than one individual, like there could be bunches of halorubrums, right? And they all have a unique gene too. So you could actually end up with hundreds or thousands of distinct individual 16S genes. And you could clone them all in theory. You could come up with 1,000 clones and you could sequence them all. And so each one should have just a single copy that ultimately came from a single cell that you lysed in your extraction procedure. Does that make sense?
Yes, thank you.
Okay. And so each unique clone then becomes like a little factory making lots of copies of that individual gene of interest. And remember, this is just 16S as an example. You can do this with any gene, right? So geneticists don't do what we do as microbial ecologists, but they use cloning all the time. Like maybe they're interested in studying some gene in a plant or an animal or a microbe. And when they clone it, they're putting that into E. coli so they can then get the sequence and study that gene of interest. Okay. All right. And we use antibiotic sensitivity here, but we're not studying antibiotic resistance in bacteria using this method. Okay. So the best answer was really B. Okay. Does everybody see that now? All right. Okay. Okay, so in the old days, we used to use cycle sequencing, which is referred to as the Sanger dideoxy method or the Sanger method. And so this method was used for decades. It's still used, and if you want to sequence clones, or let's say you have a pure culture and you want to sequence the 16S gene from that pure culture, you can still amplify the 16S genes from it, send it to now we usually send it to a company because sequencing companies now abound and they're very cheap. And they can use this Sanger method. What this does is it either is off of a plasmid or it's off of a PCR product. And they run another PCR reaction with it. Okay. But it's not exponential like the PCR reaction we've seen, right? So you don't have the S curve. It's a linear reaction where basically you don't need to make lots of new copies of it. What you need to do is you need to insert fluorescently labeled nucleotides. And so when I say DNTP, that refers to deoxynucleic acids. And it's a mixture. When we see N, that's a mixture of A, C's, G's, and T's. Okay. So all 4 DNA nucleotides are represented. And so what they do is you're running a PCR reaction with just a single primer, and you are sequencing the complementary strand, okay, off of one of your products that you send them. So you've got millions of copies that you send them in the mail, and then they add it in a tube and they do this reaction where they're trying to fluorescently label it. And so every time there's an A put in there, it's a single color, and a C and a G and a T. But we don't label all of the nucleotides because that wouldn't really help us. What we do is we label 1% of them. And instead of being deoxynucleic acids, they're dideoxy or DDNTPs. And what happens is when Taq polymerase inserts one of these, let's say ATP, DATP, it sticks the A in and then it grabs another nucleotide and goes to the next and the next. But if it grabs a dideoxynucleic acid base and sticks it in, that stops Taq polymerase in its tracks and it can no longer synthesize the complementary strand. Okay. So ultimately, this is a random process. Taq is not smart. It doesn't know whether it's a DNTP or a DDNTP. So it is just grabbing what is in the tube in the environment around the enzyme, and it will just have a 1% chance of grabbing that DNTP for any base as it progresses. And so what happens is you end up with different fragments. You're doing this thousands to millions fold. So you get lots of fragment sizes. And that's shown here. So here's the primer. And then this is a case where it stuck the DDGTP in really quickly. So it just by chance grabbed a dideoxy one at base like 2 or whatever that is, base 1. In this case, it made it to base 2 and then it accidentally grabbed that. In other cases, it may make it to base 400 or 1,000 before it grabs a dideoxy one. And so what you get is a whole mixture of succeedingly longer and longer and longer fragments, and all of them stop with one of these dideoxy ones. But what do you notice about these 4 different bases, the T, C, A, and G?
They're tagged.
And what's different about it?
Well, there's different colors for each one.
Right. And so in this case, that symbolizes the different fluorochromes. There's that word again. We can use it under the microscope, but we can also use it with these lasers. So what you do is you have a different color, and then you run this mixture through a gel. And in this case, what we do is we use a little tiny capillary filled with just a very small amount of gel. And in those gels, those are not agarose gels that you may have run. These are acrylamide gels, polyacrylamide. But regardless, what happens is they run through this. And just like with an agarose gel, when you run it, the smallest things run fastest through the gel and big fragments run slowest. So this guy will zip right through, but something that's like 200 or 300 base pairs long, right before it had the base entered, will take a lot longer. And so what happens is you run it through the capillary and that separates out. And then there's a detector at the end, and the laser flashes every time. And so the first one to flash will be purple or pink, I guess it is here. So it went pink, pink, red, blue, green, red. And it just, it's hooked up to a computer, right? So it logs all of this. And um, this is what you end up with as a product. So each peak represents a flash, and the color corresponds to the different nucleotide that was the dideoxy that got inserted. Does that make sense? So that is how Sanger sequencing works. Did anybody here know how that worked before today's lecture? Probably not. So I know it's a little esoteric. I'm not going to be testing you on how Sanger sequencing works in great detail or anything like that.
I think we did
But
study this in the journal bio, but it was just like mentioning
right.
no video about it.
Yeah. And, because we can't have labs right now, I like to give you like some sense of the fact that this is what actually happens inside a laboratory and this is how that works. Instead of just giving you the name of it and moving on, now you know a little bit about it. All right, so then you get your sequence. And this is what they talked about in the paper. This is actually one of my own. I won't tell you when I did this. I don't think there's a date on here, but it was a long time ago. This is one of my own 16S sequences that I got. And what you do is you take your sequence that you get back from the sequencing facility and you blast it, right? And when we blast, what are we doing? Who knows?
Checking what other organisms might have the same sequence.
Yeah,
That's
so yours is the query, basically like a question, right? You're asking a question. This gene sequence, what's it similar to? And if it doesn't come back as similar to 16S gene, you know something went dreadfully wrong with your with your procedure. But assuming it did, then what you're looking for is something that is high similarity. And that's determined by this E value. The E score, the lower it is, the better off it is. So when it's 0, it's good. And so what this showed me is that this clone sequence was from a cyanobacterium called Microcoleus. So the things that were in the database. And so I knew that this was a cyanobacterial sequence because it's essentially identical. So this is very similar to what they did in the Oxenwright paper in terms of doing a preliminary identification. But then ultimately you want to make a tree, right? And so this is what they did as well, but you didn't get to see what it looks like. So these are some sequences. These are actually not 16S sequences. These are protein coding genes, and you can tell that based on the names or whatever. But essentially what it is what we want to do is line up the sequences so they all start with the primer site. That's where you should be starting, right? And then you need them to line up so that base number 10 is lined up with base number 10 of the other organism, right? The other sequence, the other species, so to speak. But guess what? Sequences vary, as you can see here, right? So here it's a T. It was written as a U, but you can ignore that, right? It's a T, but it's a C in this species. Okay. Sometimes 4 of these have the same thing, and this one is different. Okay. So you're going to have nucleotide variation, and you're also going to potentially have insertions or deletions in the gene, right? And that's what's represented here. This is a 1, 2, 3, 4, 5, 6 base pair indel, we call them, right? In this case, it was probably an insertion in this species because everybody else is missing it. And when it's 3, 6 bases, this is a protein coding gene. So that means it had 2 extra amino acids if you translated this, right? So alignment becomes very important because you may have varying levels. Now, 16S genes are not protein coding genes, but they are variable in length. They have this interesting, because it's RNA, they have like stem loop structures and some interesting things that they form inside the cell. And so you have to align all genes, no matter whether it's RNA or DNA. Once you've aligned them, and usually we use computer algorithms to do this, okay, then we're going to plug them into a phylogenetic tree method. And this is also going to be computerized. And there's a few different methods. I'm not going to go into all the details because I don't have time to do that. But what I want you to appreciate is that there are different methods that can be used to make trees. So the phylogeny that was made in the Oxenwright paper was made, I believe, via neighbor joining, if I remember correctly, which is one very commonly used method. Phylogenetic trees, as you saw, though, are not they give you a tree. You run data aligned sequences through a program, it's going to give you a tree. But one of our important questions is the tree real? Right? Does it really reflect the evolutionary history as it should? Because if you think about it, every one of those nucleotide base pair changes that we saw here, right? These arose via a mutation in that lineage that didn't occur in the other lineage, right? So all of these guys have the same thing. And this one had a mutation that changed the T or the U to C. Okay. So those mutations are a record of the evolutionary history of that gene sequence or of that genome, right? Of that individual organism. And so it turns out that the model of evolution that you use, and that's what these are. Parsimony is one method, which basically says we want to figure out the fewest number of changes we need to make in the data set to account for all the differences we see in those nucleotide changes. That's one model of evolution. Neighbor joining is another model. Maximum likelihood is a probability-based model. All of these are different approaches and none of them are perfect. And so the takeaway from this is that no one method is likely to give you the perfect phylogenetic tree. So it's always a good idea to use multiple methods if you're really interested in understanding the evolution of a gene sequence or of species. And one thing we can do also within a method is we can run it over and over and over. So I can make the tree and then I can make it again and again and again and again. And that's called bootstrapping. It's not really replication, it's more pseudo replication because we're essentially taking the same data and we're analyzing it over and over and over, but we're not adding new data. So technically it's pseudo replication, but for our purposes, we can just think of it as replication. And so what you can see here, this is like one of my old trees. And the percentages here are the percent of time that we see the branching pattern in the tree this way. So if I run the tree, 67% of the time, this, and this came together. That means that a third of the time, maybe one of these guys was down here and grouped with these organisms in the tree. So ultimately, we'd love to get 100% bootstrap replication, and that would give us really high confidence. And sometimes you come close, right? Like this is 98% of the time, all of these guys clustered together, okay? So it gives you a sense of how reliable your data are. That's what bootstrapping does, okay? All right, so the take home is we can reconstruct these trees. We can get a sense of identifying, which is essentially what the halophile paper was doing, right? They were trying to figure out, okay, so do we have Halorubrum, Haloarcula? Who are we missing? There's some cultured guys, there's some uncultured guys. We wanted to compare those. These are the methodologies that they use to generate that. So hopefully this is like sort of, I've pulled the emperor's clothes off and you can see kind of like how this all works. You may not have understood that as we were reading the paper, but does this make things a little bit clearer on how they did that in the Oxenwright paper?
Very.
All right. Are there any questions?
Did you use a maximum likelihood tree on or
So this
Almost.
actually, I can't remember exactly this one, but typically when I made trees and like published the phylogeny, I would use all three methods and I would compare those three trees. And so I would put indicators on there. Oh, I saw this branching pattern. So oftentimes in papers, you'll see like symbols. And so like you put a black circle every time that there was high support using all three methods or something like that. And you would just have to put in your figure caption for the paper. You'd have to report what that means. But yeah, you never want to publish using just one method if phylogeny is your main point, right? Which it really was in like the Oxenwright paper. So I would have preferred to see them use multiple methods. But anyway.
When do you know if a tree is like rooted? Like, I think theirs wasn't. That shows like
Right.
ancestry, if I'm
Yeah,
not
so
mistaken, right?
technically, rooting is a whole other thing that we don't really have time to go into, but you can artificially root. And so what I did here, these are all bacterial sequences, and this is an archaeal sequence. So these are actually not 16S genes. These are sulfite reductase genes, which I isolated from these hot spring sulfate reducers. Anytime you see desulfo or thermodesulfo, that it's a good indicator it's a sulfate reducer. This is an archaeal genus that also has that gene. I got it through lateral transfer. So I use this as what we call an outgroup. And the outgroup can be used to help to root the tree. Because essentially what we know then is everything here is bacterial and with reference to this one archae. But if you're doing like the tree of life with ribosomal RNA genes, you can definitely play around with that. But doing true rooting is actually really complicated, so most people don't do it. That's for like the really hardcore phylogeneticists. All right.
Gotcha. Thank you.
Yep. So that is old school cloning and sequencing, etc. But now we're new school, right? So now we use all these fancy new methods that have been developed, which really came about because the Human Genome Project forced scientists to develop high throughput sequencing approaches. And then companies got involved, and now we have commercially available rapid next generation sequencing. Same thing is done. You still have to extract your DNA or RNA from your samples. But remember all that cloning stuff we had to do before? We don't have to do that with new sequencing approaches. Now, there are a number of next generation approaches. And in fact, the Chivian paper used a method that is no longer used because it's a little more error prone. But that's okay. It's still reasonable data. But these days, Illumina, which is a company based out of San Diego, you may have seen it if you drove down I-5. It's right off the freeway there. So Illumina has kind of, at least for now, won the sequencing battles. And so their approach is very rapidly being used by lots of people for genome sequencing. There are some other companies like PacBio, which is from NorCal, that also are making headway. And they have a different technology which generates long reads. Illumina, one of the downsides of that is it has short reads. Okay. The one you'll read about in Chivian is the 454 method, which was another company. All of these end up generating what we call contigs. And essentially what you're doing is you fragment the genomes and then you've got to reassemble those genomes, right? So you break the genome up into a bunch of chunks and then you've got to figure out like a puzzle, like where they go back together. And so what you need are overlapping regions where the sequence would be the same here as it is here. And of course we do this with a computer. This is fairly simple to do with a pure culture genome, right? So you've got different fragments, you assemble them in the computer, and you end up closing the genome, so to speak, which means you get sequences that overlap all the way around, assuming you have a circular prokaryotic genome. And so what we do is we sequence a lot, and that's the really great thing about next generation sequencing is, with clones and Sanger sequencing, you would get hundreds of reads. With next gen sequencing, you get millions of reads or tens or hundreds of millions of reads. And so what that allows you to do is get very high redundancy in the sequencing. And so we call that coverage, right? So how many, in this case, we only have two-fold coverage or one-fold coverage really, but you can end up with lots of different reads. So maybe we have 1,000 of these and 1,000 of these and 1,000 of these, and you can line them all up and compare them. And that helps you to, that redundancy helps you to reduce errors. All of these methods have, they're PCR based. And so all of them end up making mistakes. The more copies you have, you can compare the sequence, and maybe there was an A that accidentally got inserted in one read. But if you have 999 reads that have a C at that position and one has an A, we would just throw out the A. Okay. And so that helps you to have accurate sequences that overlap. Okay. All right. But this is tricky when you have mixed samples. And this is like the example of the Oxenwright paper. So the Oxenwright paper had a mixed set of genomes. They used PCR, and then they used cloning to separate out the genes. But they were only looking at one gene, right? What if you want to look at all the genes? It's really tricky with mixed samples. But this is what is essentially the fundamental basis of metagenomics, which is the methodology that's used widely now in microbial ecology. And bioinformaticians like Dr. Berlimon in our department, this is their bread and butter. This is what they do. They use computer algorithms to analyze sequences and try to figure this all out. And so what they do is they have to compare to look for overlapping regions, but they have to do it with overlapping across lots of different genomes. And so you end up with situations like this. And so you have to use computer algorithms to figure it out and separate the pinks from the grays, from the reds, from the purples, from the blues, whatever. But it can be done. And it is done very widely in the field of metagenomics. What this allows us to do is to get large numbers of sequences, millions of reads of sequences, and separate them. The reason we don't have to do cloning is that these methodologies have a different technology for separation. And essentially what they do is they take the mixed double-stranded DNA, you add it in. You're not going to be tested on this, by the way. Okay. But I want you to see how this works. And you can see that you add these adapters. The adapters stick onto a plate. And then what they do is they run PCR on this plate in high redundancy, high throughput. And similar to what we saw with the Sanger, they use different colored dyes, different fluorescent dyes for the nucleotides. But instead of running this through a capillary and flashing it with a laser there, these are all stuck here and they just propagate and replicate next to each other. And then the laser scans over this entire plate. And so it will detect, oh, there's a bunch of these that ended up with the terminal base being the C. Okay. And so they still have that kind of fluorescent labeling that goes on similar to what we saw with the Sanger sequencing, but you don't have to do cloning first. You just run it through the machine, have it stuck on these plates. And then what you can see here is it gives you these sequence reads. Okay. And those correspond essentially to, oh, so there's that base and that base and that base. And so the computer assembles all of the nucleotides based on the fluorescence. Okay. That's about as far as we need to go with that. But the thing you really need to know is that there is major high throughput that you can achieve here. And it's very reliable. But the problem is you end up with these short little fragments. And so that's why some of these other technologies like PacBio are also going pretty well. All right, so we are over time. So we will pick up with this next time, and then we'll talk about quantitative methods, and we'll probably be able to start the activity measures, which is a shorter lecture, but we may have to finish activity in our last lecture before the midterm, which is also a question and answer session. But that'll all start next Wednesday. Remember, Monday, we're talking about the Chivian paper. So be sure to write those. Look at your old summaries, write your new summaries. If you have any questions, just let me know. And we'll have a discussion on Monday, just like we did with the Oxenwright paper.
Thank you, Professor.
You too. Have a good weekend.
Dr. Dillon,
Yes.
you're saying that to access the notes, we have to do what on the Word document? I'm sorry, because I can't see.
Okay, so you have your Word document open?
I could open it real quick. The
Yeah,
one,
open it up and turn on track changes. If you do that, can you see there should be a bunch of comment bubbles on the side? Okay, Robin, you said you found it. Did you find it, Daniel?
I'm doing it now.
Oh, okay. So Robin, you were able to see those comments on the side then? Okay, cool. All right, so just read through those and use that feedback for writing yours was pretty good, Robin, if I remember. So yours just had a couple nitpicky things.
Thank you.
Yep. See you on Monday. And if I remember correctly, Daniel, I think yours where you were missing some of the responses. That was where you got dinged at times, but I can't remember. I graded so many of them. It's hard to remember.
No, I understand. You know, is it?
Can you see it?
No, it's not coming up. That's what I'm trying to figure out.
Okay.
Because I'm on my
Let me. I can try just emailing it. Let me just look at the file to make sure it didn't get somehow magically lost. I mean, I know I saved it.
Okay,
But
you know,
you never know.
I was Dr. Dillon. I was downloading the wrong one.
Oh, yeah, you have to get the one that. Okay,
Yeah, I got it. Thank you.
you got it? Okay, cool. Sounds good. Erica, did you have a question too?
No, thank you.
Okay, I'll see you on Monday then.
Yamane.
Okay, bye.